Our next speaker is Dr. Ratliff. You know her, so I don't have to say more about her. She is my colleague in Homeland Security and Materials Management Division within CSER ORD EPA. She is a principal investigator within the EPA HSMMD, working under the HSMMD Homeland Security Research Program. She uses numerical models, lab and field scale studies to develop and evaluate tools for remediating environmental contaminants, including leading EPA's research to evaluate efficacy of different air treatment technologies against airborne pathogens. She received her BA in Earth and Environmental Sciences from Vanderbilt University and a PhD in Earth and Ocean Sciences from Duke University. And you have heard her name and many publications. She is co-author and done extensive work. Welcome.
Thanks, Sanjeev, for that nice introduction. And I want to start out by acknowledging my co-authors, many of whom are sitting here in the audience today. And also want to state that this work is all part of a broader EPA project team supported across multiple offices. So I want to acknowledge the hard work that's gone in from a number of different people. So probably don't need to spend too much time motivating this. As a result of the COVID pandemic, I'm sure many of you all are aware, but there's many different types of air cleaning and treatment technologies that are on the market. So when I say air cleaning technologies, I'm typically referring to those that are capturing and removing particles from the airstream. And when I talk about treatment technologies, I'm typically talking about things that inactivate microorganism in the air, but I'll be using these terms interchangeably. So there's things that emit ultraviolet light, ions, all those types of things that would potentially inactivate infectious aerosols, but again, also filtration-based technologies that would remove those infectious aerosols from the airstream. And then there's also all kinds of combination devices, so things that include a filter plus UV or filter plus ionization. And as there was a growing concern and recognition of the airborne transmission of COVID-19, we started getting all kinds of questions from different stakeholders asking us what sort of technologies can we use in our particular spaces, whether it's a classroom or a subway car or a bus, to reduce the risk of airborne transmission of COVID-19. And all these questions were coming to us as we were trying to reoccupy those indoor spaces and do that in a safe way where we were reducing that risk of disease transmission. And as we started to dig into those data that back up the efficacy studies of these different technologies, we realized it was really hard to understand and wrap our heads around those comparisons between the different types of technologies. There's a big lack of standardized testing for these kinds of technologies, which makes it really difficult to compare across the different technology types, or even within one type of technology, for example, UVC, things can be tested in very different ways. So it made it really challenging for us to provide that kind of expertise and support to those stakeholders and address those questions if we don't understand how to draw those comparisons. And so within the Office of Research and Development, we developed a new program to start to address these questions. So fundamentally, we want to understand how effective are these different technologies in reducing the concentrations of infectious aerosols. How can we start to draw the comparisons across the different technology types? And then how can we take what we find in the lab and extrapolate to the real world? So another thing to note is that a lot of these technologies are tested under highly idealized laboratory conditions. So the conditions under which they're tested may not be relevant at all to where they would actually be applied, which again makes it challenging to address those kinds of questions about what should we use where. Already sort of talked about some of these challenges, but again, want to note that I'm not going too far into the regulatory weeds, but the vast majority of these technologies are technically considered pesticide devices. They are regulated by EPA under FIFRA, these devices, but EPA doesn't register them, meaning they don't go through any sort of pre-market review in terms of the efficacy data. So regulated but not registered, EPA doesn't review those data, which again can make it really challenging for us to understand how those data are relevant to the application. And again, a lot of the work that's been done on these devices is testing that's been done by the manufacturer. So no big surprise that manufacturers want to test in a way that makes their device or technology look good. So again, we developed this research to start to address these challenges, want to understand how effective they are. These technologies are under obviously laboratory conditions, but conditions that are also more translatable to the real world. So I mentioned again that these technologies are often tested in a lab under conditions that are favorable for the device. Think about an air cleaner in a glove box. That's how a lot of these things are often tested. There's not a lot of air to clean. So if we take, for example, log reduction data from these type of tests, it can be really hard to take those data and extrapolate them to a classroom or even an auditorium, which is where a lot of these devices would be actually used. So the data that are generated in a glove box are challenging to extrapolate and predict performance in those sort of more real world settings. So if you take away nothing else from the talk today, I want you to, wrap your heads around the fact that scale matters. We know that for a lot of the work that we do and a lot of work that's been discussed already, but for air related research, it's really, really important. And so to overcome some of these challenges, we've been using a relatively large bioaerosol test facility in our RTP lab. We have 3,000 cubic feet of space in a bioaerosol chamber to conduct these kind of tests. We've constructed a mock HVAC system that has been designed to broadly simulate a range of different indoor environments. And we use temperature and humidity controls to control the environmental conditions within the chamber. So we've been using primarily the bacteriophage MS2 as a surrogate for SARS-CoV-2 and other infectious aerosols. It is a BSL-1 microorganism, so it's safe to work with, which is really important when we're doing this kind of research at large scales that we want to be doing it at. We aerosolize and another thing to note about MS2 is that it's a small non-enveloped phage. So it means it's going to be more resistant than SARS-CoV-2 and other coronaviruses. So importantly, if something works against MS2, we have a lot of confidence that it's going to work or inactivate SARS-CoV-2. So we aerosolize the MS2 into the chamber, and I'm going to be talking today about aerosolizing using two different types of media. Aerosolizing using deionized water in the nebulizer compared to simulated saliva. And what I'm going to be, again, talking about in some of those results is the differences between the two, because a lot of the testing that's been done is, again, done under these highly idealized laboratory conditions with, say, for example, deionized water as the media, but simulated saliva is more representative of those real world conditions. When we as humans are emitting infectious aerosols, it's not naked virus. It's going to be coated in mucin and salts and other types of proteins that make potentially inactivation more challenging. So we want to account for that in our testing. So we aerosolize the microorganism and we also sample the air during a test, and we take those samples back to our on-site microbiology lab, and we enumerate how much infectious aerosol is present in the air over time throughout testing. And we also have some particle sizing instrumentation. I'll be talking about data from an SNPS, a scanning mobility particle sizer, and an APS. The SNPS sizes a smaller size fraction, and the APS is used for a slightly larger size fraction. And I'll be talking in a minute about why the replicates are important for testing. So how do we calculate or quantify efficacy? The main key thing I want you to take away here is that regardless of how we talk about efficacy, we're always talking about it relative to a control condition. So that's really important. I don't really need to motivate that for, I think, most of this audience here, but those control tests where we don't actually have the technology on are really critical to quantify that background amount of microorganism decay that would be happening without any technology operating. So by using those control tests, we're able to look at the change between how much decay we see with the technology on versus the technology off, and truly isolate the effectiveness of that technology, not just the overall reduction of the microorganism. So log reduction is something that's probably familiar with many of you all in the audience. We can calculate log reduction based on time-matched control tests compared to the technology tests. But something that may not be quite as familiar to many of you is calculating a clean air delivery rate. So we can look at comparing the first order decay rates of microorganism with the technology on and off, looking at the difference between the two, multiplying that by the volume of the test chamber. And that gives us essentially a quantification of how much pollutant-free air is delivered by a technology over time. So this is something that is specific to the pollutant. So I'll be talking about a few different clean air delivery rates here: an infectious aerosol or MS2 clean air delivery rate, and the two different particle sizes. And so the whole point of my talk here today is to tell you and show you how the results that we generate and the efficacy data that we generate are a function of the test methods that we use in the study. So a couple different devices and tests I'm going to talk about today. First being a far UV device. So this is a type of device that's going to be inactivating microorganism in the air. And there's been a lot of excitement and talk about 222 nanometer UV over the past few years. It's an emerging technology. That shorter wavelength is less damaging to human skin and eyes than the traditional 254 nanometer wavelength UV that's often used or been used for decades. And so as a result, it's actually being used in occupied spaces now. And so we used two of these devices in the test chamber based on recommendations from the manufacturer, and tested again with deionized water and simulated saliva as the aerosolization media. And the graph that I'm showing here has time on the x-axis and the concentration of microorganism on the y-axis, and each of these lines represents a single test. So the gray dashed lines are the set of control experiments, and then the orange lines are the experiments where we had the far UV devices on. So the first thing that I want to point out is that for just the control tests, we saw over a 2 log or a 99% reduction in the amount of MS2 present in the air, even without the technology operating. So when we talk about, again, quantifying efficacy, it's really important to have good time-matched controls. Otherwise, we could be overstating efficacy of the technology for this particular example by over 99%. The other thing I'd like to point out is that the variability in MS2 from a given sampling time point here ranges over a log for some of the samples. So that means that we need to be conducting replicates. There's a lot of scatter in the data that's just inherent in working with microbiology or microorganisms, but particularly inherent when we're talking about aerosolizing microorganisms and trying to recover them. So again, replicate testing is really, really important for characterizing efficacy. So this top graph here is now just averaging over those replicates and normalizing those replicate tests and normalizing. And then the bottom here is a bar graph showing the log reduction at each of the specific sampling time points. And so we saw up to about an 83% reduction at 90 minutes with the far UV device on and attributed to the far UV device, with statistically significant differences between the control and the test conditions at 60 and 90 minutes. So now what happens when we aerosolize and simulated saliva? Well, we actually get some interesting, somewhat counterintuitive results. So if you look again, the orange line is aerosolizing in simulated saliva, and the purple is aerosolizing in, the orange line is aerosolizing a DI, and the purple is in simulated saliva. So there's more overall inactivation of microorganism in the deionized water. But the really interesting thing is here that the control, this sort of darker black line, is the control with the simulated saliva as the aerosolization media. So there's better bioaerosol stability. The MS2 is happier over a longer period of time in simulated saliva. And since the log reduction is the difference between the control and the test, and that's how we're calculating efficacy, there's actually a greater difference between the control and test for simulated saliva as the aerosolization media. So a little bit counterintuitive in that more overall microorganism reduction in DI water, but the calculated efficacy is higher in simulated saliva. So now moving on to look at a different type of technology, a portable HEPA air cleaner. So something that's going to be capturing and removing particles from the airstream instead of inactivating them. And we use 2 of these air cleaners in the test chamber based on the cleaner delivery rates and how you size them for the space. So same type of results for this particular set of devices. For deionized water, we see up to a 90% reduction in infectious aerosol over the 90-minute test period, with statistically significant differences at 30, 60, and 90 minutes for aerosolizing and deionized water. And with simulated saliva, we see up to a 99% or over a 2 log reduction at 90 minutes. And it's the same story. We see more overall reduction. This green line is aerosolizing in DI. The blue line is aerosolizing in simulated saliva for the HEPA tests. more overall reduction in infectious aerosol with deionized water, but the difference between the test and the control is higher with the simulated saliva. So we see a greater calculated efficacy there. So this, I'm not going to spend too much time here, essentially summarizes what I've been saying over the last few slides. There's a greater overall reduction in over the test period with DI water compared to simulated saliva, but when we look at the time-matched controls, that flip-flops with simulated saliva being more effective. So how do we translate this into a clean air delivery rate? The dots here are the same data that you saw before, and the dashed line now is a linear regression fitted through those data. And we can calculate the clean air delivery rate as the difference between the slopes and the lines, essentially calculated, multiplied by the chamber volume. So the clean air delivery rate that we get for far UV devices, and again, reminder, it's 2 of them, is about 62 CFM, and for the HEPA units, about 100 CFM. When we look at calculating the clean air delivery rate with simulated saliva, that actually doubles. So changing the aerosolization media essentially doubles the calculated efficacy or the calculated clean air delivery rate. So again, reminder, 2 of each of these devices in the chamber. So clearly this aerosolizing and simulated saliva increases the stability of the bioaerosol, and that's driving those differences in what we see calculated, whether it's the log reduction or clean air delivery rate for efficacy. But this begs the question, is there something different going on with particle dynamics when we aerosolize and simulated saliva compared to deionized water? So now these 2 graphs are the mass weighted particle size distribution for the 2 different aerosolization media, and the mass median diameter is the same. So we're not changing the median particle size by changing the microorganism suspension. We can also look at, where is the infectious aerosol actually located across that size range. We have an impactor that allows us to take samples at different size cuts and enumerate at those size cuts. So we can look at how much aerosol is located across the different parts of the particle size spectrum. We see for deionized water, there's a sharper peak at about 0.3 microns. And then for simulated saliva, it's a broader distribution where it's sort of ranges between 0.3 and closer to 1 micron. So we are changing where that infectious microorganism is located, even though we're not changing the actual particle size the particle size distribution itself. So this suggests that we need to be considering a broad range of particle sizes and looking at, is there more capture in one particle size range over another? So now these data are showing the particle size concentration for the smaller particle size fraction, so less than about 0.6 microns, and over the test period. So again, test period on the x-axis here, and the particle size concentration on the y-axis, and on the graph on the left is the deionized water test. The graph on the right is the simulated saliva test. They look the same. The amount of particle reduction as measured over the size range is about 97% over the test period. And we don't see, and that's for the HEPA unit, I should say. So you can't actually really see the far UV line because you can't really see the control line because it's essentially overlapping with the far UV in both of these scenarios. And that makes sense. We know UV isn't capturing and removing particles from the airstream. The HEPA unit is. There's different mechanisms by how these things are removing or reducing that infectious aerosol concentration. But long story short, we're not changing the capture or the particle size dynamics by changing the aerosolization media for this size fraction. For a slightly larger size fraction, for 0.5 to 1 micron, the story is the same. We don't see any reduction for far UV. We see about a 98% reduction for both of the aerosolization media for the HEPA units under these conditions. And so we can also look at, calculating those clean air delivery rates for the particles. Again, no big surprise. We don't expect to see any reduction from far UV, but we do see that from the HEPA air cleaners. This calculated clean air delivery rate is about 135 CFM for both the DI and the simulated saliva aerosolization media for that smaller size range, the SMPS measured particles, and it's about 20 CFM higher down here for the APS, but it doesn't change whether you're aerosolizing in simulated saliva or deionized water. And I will note that it makes sense that the slightly larger particle size range, the APS measured particle sizes, have a higher capture efficiency because this smaller size range, the SMPS, contains the most penetrating particle size for filtration, which is around 0.3 microns. So that size range is harder to capture in a filter than the slightly larger particle sizes. So these data suggest that changing the aerosolization media isn't changing those particle dynamics for our particular test setup. So this just summarizes all of these results, the cleaner delivery rates, where we see again that the simulated saliva compared to the DI water changes that microorganism cleaner delivery rate for both the technology that inactivates and captures, but nothing is substantially changed for the particle size particle dynamics. So to summarize, we have shown that something as simple as changing the aerosolization media can have this significant impact on the calculated efficacy. We see more overall reduction for both conditions using deionized water compared to simulated saliva, but that calculated efficacy is actually greater with simulated saliva because of the enhanced stability of the controls. That's really driven by the microbiological dynamics, not the particle size dynamics. Again, I want you to keep in mind that those time-matched control tests are really critical for calculating efficacy, and we see more variability in the bioaerosol results than the particle size data. And so, a few takeaway points here: that standardized test methods are really critical for understanding how we can compare and contrast these different technologies and how we translate results from the lab to the real world. And under these particular conditions, the HEPA units are more effective than the FAR UV units. And just as a quick side note before I wrap up, why do we care about all of this? Well, there's new public health-based targets for ventilation that are being published. The CDC, for the first time, released a health-based ventilation target earlier this year of 5 equivalent air changes per hour. Air cleaning can be part of the way that you get to that 5 equivalent air changes per hour, but they don't specify how you test for it. So the data that we're showing here suggests that even something really seemingly minor can have a really significant impact on how many, say, UV devices you need to deploy, or HEPA units you need to deploy in a space to get to that target air changes per hour. And the exact same thing for the ASHRAE 241 control of infectious aerosol standard that was published earlier this year. Air cleaning is a way you can get to the targets that are published in that standard. And there's significant advances that are made in that standard for standardizing the way that air cleaning technologies are tested, but aerosolization media isn't specified in there. So just want to make sure everybody, has that in the back of their minds when they're looking at the efficacy data for these technologies. And with that, I'm probably out of time for questions, but thanks for your attention.
Questions?
Thank you, Catherine. A very good talk. Question for you, 2 questions actually. First, do you have some idea on decay curve when MS2 is dried at its spot versus MS2 release as aerosol? In both conditions, they're decaying, but is it faster one way, or they're similar, or they're different? That's the first question. The second question is the FAR UV. What is the distance where the source is to the MS2 and relative energy levels or dosage? Thank you.
Yeah, so to your first question on the decay curve, there's a number of studies that have come out of like Lindsay Marr's group that have looked at this. It's not something we've focused on in particular, but we've seen that at lower humidities for the aerosol work, we see greater bioaerosol stability. In MS2, there's actually a saddle in the viability. So around 50% RH is the lowest stability, and at lower RH, it's higher stability, and higher RH, it's higher stability as well. So, but that's going to be microorganism dependent. And I haven't done studies to really compare that to service-based work. And to your second question about the FAR UV units, we consulted the manufacturer and how we deployed them in the chamber. We essentially deployed them on the ends, the short sides, you've seen it, pointed inward to try to minimize any shadowing in the chamber. And I think it's 14 microwatts per centimeters squared at 1 meter centered distance from the units. And they're the commercially available far UV unit that's most widely purchased across the world. So, but I'm happy to talk to you more about the specs for those, and you're welcome to borrow them. Yeah.
Hi, I'm Chris Bowers from Exemer. I was wondering, I've got two questions. One is that I'm sure you've thought about this, that when someone's infectious and they're in a room, they're continuously breathing out infectious particles. Have you thought about doing any experiments where you're doing kind of a steady state continuous injection into the room and seeing how the performance of different technology compares to the control? And then the second question is, for the filtration, the HEPA filters have a fan in them, and it seems like you're getting some loss due to settling. Have you tried to see how the HEPA filter impacts the loss just turning on the fan and not having a filter inside?
So great question about the filter. We have done some testing where we've looked at like installing a filter or not in the HVAC system, and based on the particle sizes that we're generating, the settling is minimal. We might get some wall losses just as particles interact, and also agglomerate within the chamber. And then, so that's something that, you know, is the back of our minds, but I don't think significantly impacts our results. And then remind me, Rogan, so I target it quickly, the first part of your
Yes. Have you thought
the
about doing
continuous release.
continuous release?
Yes, so we actually just started running those experiments. So I'm really interested in that. The way that we designed these initial experiments, we were hoping to get that real high bioaerosol pulse into the system and run those decay type tests, which are more consistent with how, say, pesticides are typically tested. But we recognize that the continual release scenario is more relevant to you know, a viral shedder in a space. And so we're excited to do those tests as well.
Thank you.
Yeah, thanks.
Any other questions? So Worth, I'm adding a comment again. Why do we care? We care about this research because it opens the door for future research under the One Health program. And our director, Lance Brook, is leading the environmental sector under the One Health program. And we work at interagency level under that program. And this kind of work lays the foundation for future and potentially real-time research. Thank you.